Okay, so good morning, everyone, and thank you for having me here. And my name is Pamela Rivera, and I studied a bachelor's degree in chemical engineering at la Universidad Autónoma de Santo Domingo in the Dominican Republic. And right now, I study a master's in analytical chemistry at the University of Tartu. And I am doing a mobility program in the University of Claude Bernard Lyon in France for a specialization in industrial analysis. So the topic of my internship was generality of the relationship between SPAD values and leaf chlorophyll content across different fertilization treatments in wheat and barley. And I was under the supervision of Leah Halleck. So here is a brief overview of this presentation. We have the objective, chlorophyll and why measure it, SPAD as an instrument, and we will give a brief explanation of how does it work, method, results, and conclusions. So our objectives were first to study the effect of species and fertilization on leaf chlorophyll and nitrogen contents, describe the covariation patterns of other leaf traits with SPAD values and chlorophyll content, test the universality of the prediction formula to estimate leaf chlorophyll and nitrogen content from SPAD values at contrasting fertilization levels, to explore the suitability of linear and exponential relationships for prediction, and provide a common prediction formula for estimated leaf chlorophyll from values in Estonian main crops for various soil fertility conditions. So first we have chlorophyll and why to measure it. And chlorophyll is a green photosynthetic pigment which helps the plants to get the energy from light. But measuring chlorophyll in the plants can give us a general overview of the health state and can help us determine the nutritional status and if there is presence of any stress factors that could affect their development. And this information can be used to improve crop yield and to also can be used as the input for the management of fertilization programs, also for programs of the application of pesticides and herbicides. Okay, one second. Sorry, I am back. So also from the measurement of chlorophyll, we can get an approximation of the nitrogen content that is present in the plants. And this is very interesting due to the role that nitrogen plays in the plant's development, since it is since it has a major effect in the photosynthesis process, since it is a major component of chlorophyll in the catalyzation of chemical responses, because of it is also contained in the structures of the genes and in the transportation of electron process that occurs in the mitochondria, since the ATP has a good amount of nitrogen in the molecule, and also the intermediate compounds are composed of nitrogen. And the information about the information and the correct use or application of nitrogen in the field has socioeconomic impacts, since it is factor that can determine the production yield, the quality of the products, and the environmental health. So in this study, we use the SPAD, which is soil plant analysis, and this is a leaf chlorophyll method that is based on the transmittance that is obtained at in the red area of the spectrum and also in the infrared area of the spectrum. This device was developed in 1984 by Minorca, and this device has some advantages over the traditional chemical analysis since it allows us to produce immediate results that are in situ and non-destructive. And also, it does not require the utilization of solvent for the extraction of the pigments for analysis. So, how does it work? The SPAD, basically, the SPAD value is the result of the relationship between the transmittance or absorbance, 960 nanometers, and at 650 nanometers and 940 nanometers. And this, along with other coefficients that are loaded in the system of the SPAD device, is what gives us the SPAD value that we obtain as the signal after the measurements. So, to obtain the information for this study, first we did the sampling, and the species that we used were wheat and barley. And for the sampling, 4 leaves per species per fertilization program were taken. And the SPAD values were obtained in the field as an average of 4 readings along the body of the leaf blade. Then the extraction for the spectrophotometric determination was done with N,N-dimethylformamide, and 4 milliliters per sample were used. And the samples, the sample size, it was punched a circumference of 7 millimeters of diameter for each sample, and the extraction time was 4 days until the leaf tissue was transparent. For the spectrophotometric determination of the pigment, the instrument used was the spectrophotometer, and the conditions were wavelength scan modes, and then the region scanned was between 750 nanometers and 400 nanometers of the electromagnetic spectrum. And the scan speed was 1,200 nanometers per minute. And the resolution was 1 nanometer. And to convert these results that we obtain in the spectrum into chlorophyll and carotenoids concentrations, we use the Webber equations that applied for our solvent, the methylformamide, and for the resolution that at which we obtain the absorbance. Then the determination of total nitrogen and carbon was done with the instrument value mass, which works on the mass combustion. And these instruments give us the results of nitrogen and carbon as a percentage. And finally, for the validation of the regression line, we decided to test for the assumptions of the residuals for the linear models, and then it is why then validation is necessary. And then we found that in the previous while reading other articles that were written on the topic, trying to develop a calibration model for this device. The quality of the regression lines that were obtained was based on the determination coefficient, but from the point of view of analytical chemistry, the regression coefficient is not enough to say if a regression model is good or not. So we decided to take this approach to test the validity of our regression line, and to test if the residuals were normally distributed. We used the Shapiro test, and to test if the variance was homogeneous along the x-axis, we decided to use the Bush-Pagan test. And to test for the independence of the receivers from each other, then we decided to use the double Watson test. So our research, first, we have an overview of lift rates and species-specific differences. And we can see that wheat, for most of the lift rates, had the higher mean value for the different lift rates than barley. And this was only different. This was not only was not real only for the case of carotenoids to chlorophyll ratio. And I think also something that is worth noting is that the grain yield between the two species, despite having significantly different values for most of the leaf weights, but did not turn out to be significantly different between them. And then we try to describe to get an overview of the whole data, the correlation network of the leaf weights, and we perform principal component analysis, and we obtained the first principal component that explain around surface of the variance, and that was also able to separate the two species. And then this first principal component was basically dominated by SPAD values, chlorophyll, and chlorophyll and nitrogen content. But while the second principal component was dominated mainly by this nitrogen concentration. Okay. Then here we have an overview of the different leaf weights, but in this time separated by the type of treatment. The type of treatments applied were the mineral and organic. By mineral, the soils were only fertilized with nitrogen salts, while in the organic type of treatment, manure into the fertilization compounds. So we can see that organic treatment turn out to have higher values for all the pigments than the mineral type of fertilization. Then here we have an overview of the effect that the intensity of treatment had over the leaf weights, and what we say, what we describe as the intensity of treatment is the concentration of nitrogen added as kilograms per square meter in the different samples. So first we have the total chlorophyll content as a function of the added nitrogen, and in the case of wheat, the total chlorophyll content increased with the increase also of the added nitrogen. But in the case of barley, there was not really a significant difference across the three levels of fertilization that were used. Then we have the nitrogen content, again, as a function of the intensity of treatment. And we can see that in the case of wheat and barley, the intensity of treatment did increase the nitrogen content. Then we obtain the same nitrogen concentration as a function of the intensity of treatment, and this is clear to see also that the nitrogen concentration did have an improvement with the increase of the concentration of nitrogen that was added to the soil. And on this one, we have the chlorophyll to nitrogen ratio as a function of the intensity of treatment again, and just proving what we said before, that the nitrogen per area did increase with the increase also of the concentration of nitrogen added. Then we have that the chlorophyll to nitrogen ratio is decreasing as the concentration of nitrogen added to the soil is increasing. And this is true for the pool data, which contains the information of both species, and for each of the species in a separate manner as well. And finally, we compared the grain yield with the intensity of treatment. And I think that what is worth noting in this case is that the highest grain yield for the pool data and for the two species in a separate was obtained for 80 kilograms per square meter concentration of nitrogen added to the soil. So here we have taken our equation, which is equation number 4 and number 9. And then we have also plotted other equations that were proposed in other articles to describe the relationship between the chlorophyll content and SPAD values. And we can see here that the exponential equations that were proposed in the different articles, in spite of being developed for different species, then they are really close to one another, and they seem also to fit our data pretty well. In the case of the linear ones, some of them miss the target, and some of them seem to be right on with a certain range of SPAD values, but outside of that, they lose their validity. So, as said before, to perform the validation of the regression lines that we were proposing, and to see to be able to pick one or another, we did this statistical test to describe and to see if they meet the requirements that are for the residuals of a linear for the residuals of a correct linear regression model. And we can see that our regression model, linear regression model, describing total chlorophyll content as a function of a SPAD, had satisfactory results in all of them. While the regression model proposed describing chlorophyll content as a function of a SPAD, and despite of having higher determination coefficient, failed in the test that was used to determine if there was autocorrelation of the residual terms. And we did the same we also tried to provide a regression line for the nitrogen content as a function of a SPAD, and for nitrogen concentration also as a function of a SPAD. And it is interesting that nitrogen concentration actually is not well related to the SPAD value, not in a linear way, nor in an exponential way. And we can see that the determination coefficients are fairly low. And also both relationships with nitrogen concentration fail the autocorrelation test for the residuals. Then we have nitrogen content or nitrogen per area, which seem to be better explained by the SPAD value, and the linear relationship seems to be better for to describe the nitrogen per area as a function of the SPAD value. But again, this one failed the test that was performed for the autocorrelation among the error terms. And The exponential one was not also very satisfactory and had a lower determination coefficient. So finally, we have a effect of fertilization and other traits in the relationship between SPAD and total chlorophyll content, nitrogen and nitrogen concentration. And for this, we use the Spearman correlation coefficient as an indicator of the relationship between these parameters. And we can see that total chlorophyll content is very well related for the both species separately and also for the pool data. Nitrogen content is well related to SPAD values in the case of wheat and the pool data, but the relationship is poorer for the barley. And nitrogen concentration also seems to be well related to the SPAD value, but in the case of barley, the relationship is very poor. So our conclusions were, first, the speed plays an important role determining the way how the plant will react to extraneous stimuli in global context, and that will be reflected in the leaf chlorophyll content. Fertilization treatment type and intensity reflects in the leaf chlorophyll content. The exponential regression lines used to explain the relationship between SPAD and chlorophyll content derived from different species describe a similar curve and will yield similar results. On the other hand, the linear relationships are valid only in a specific range of values. And in our case, the linear regression is sufficient to generate a calibration curve to predict chlorophyll content based on SPAD values accurately, according to the results of our validation procedure. And here we have our proposed formula to predict total chlorophyll content with the linear regression and nitrogen content as well. And thank you for your attention.